Hello, welcome to Unit 1, Measuring Perceptual Processes. In the last class, we talked about perceptual processes, the seven processes. Today, we're going to talk about different ways of measuring these processes. We have learned uh, in Unit 0 that energy, the world that we experience is in energy form. The energy is in different forms, and we have organs to translate the information into our system. Light energy for vision, sound energy in case of hearing, and so on and so forth. The idea is our experience of the external energy information requires some sort of special structures in the sense organs, sensory receptors. And these sensory receptors are specialized neurons that are sensitive to energies of different kinds. For instance, photoreceptors in our eyes are sensitive to light. Sense organs in our ears have sensory receptors that are sensitive to sound. Along the same lines, we have skin receptors that are sensitive to touch, pressure, and temperature. We also have receptors in our tongue that are sensitive to chemical energy. Similarly, in our nose, receptors are sensitive to chemicals. If you look at it, our sense organs are specialized structures because of these specialized neurons called receptors. And each of these receptors are given different names. We are particularly interested in vision, and that's what we'll focus on throughout this semester. So, what do these receptors do? Well, these receptors capture the information that's out there in the external environment. And when they capture in the form of electrical signals. We experience our external world through our sense organs, but sense organs are not the structures that experience the external world. Then what are these structures? Brain is the only structure that we have that experiences the external information, meaning the light energy, sound energy, or any form of energy that we can experience. It is the experience of the brain that we feel like we are experiencing the external world. So why do we have sense organs then? Well, the sense organs are means through which our brain experiences the external world. What do these sense organs do then? Well, these sense organs, because of these specialized neurons called receptors, help us capture the external information, the energy, and send it to the brain. So what do these receptors do then? They capture the external raw energy. This capturing is sensation. In doing so, they are converting a raw energy form. In case of vision, it is light, into electrical signals. If it is hearing, your receptors in your ears are capturing the sound energy. How are they capturing? Well, they convert the sound energy into electrical signals. Take any sense organ. We have specialized receptors that capture the external energy. And this capturing is done because that external energy is converted into electric signals. And two things happen here. Number one, external energy is transformed into a different form, which is electrical signals, no matter what sensory modality we're talking about. Number two, the external energy transformation results in a representation. So that's why we have two principles here, principle of transformation and principle of representation. According to the principle of transformation, stimulus, for instance, light, and the responses it creates in the nervous system, electrical signals, these are transformed before it is perceived. And this transformation happens in multiple stages. The first transformation we talked about was a transformation that happens in the photoreceptors, which results in this capturing. But this information that is captured by receptors also undergoes changes that goes through different parts of the body and reaches the brain. And within the brain, when it goes from one part to another part. So what is transformation basically saying is that stimulus is transformed all along before it's perceived. But the first transformation results in the representation. It's like the light that is reflected from this object is captured by your retina by transformation. This transformation results in creation of a representation. So what your brain receives is this representation of this object in the external world. So representation is another important construct or principle in perception, which states that our perception is not because the light that is reflected from the pen reaches your brain and gives you the experience of the light from the pen. No. So that's why it says perception is not based on the stimulus, direct contact with higher levels of the nervous system, but on its representations first in your sense organs and the resulting activities in the nervous system throughout, referring to other transformations all along. So, what is the point about these two things? Well, the fundamental point here is that our experience of the external world are not direct, The thing that experiences is the brain, and the structures that are helping us experience the external information are our sense organs. Specifically, these are the receptors in our sense organs. It's crazy, right? When somebody touches you, when you touch yourself, the feeling of touch is not because your brain is feeling the touch, but your sensory receptor is converting that information related to the touch. That conversion happens here in your sense organ. That information is sent to the brain, and that representation is what your brain is experiencing as touch. So, our perception of the external world is limited by how effective our receptors are in capturing the essence of the external world. Well, we'll explore more later. I just wanted to give you this basic idea about it. That's what we learned. Um, we also talked about 7 stages and what how these stages can be grouped into 3 categories. And the 3 categories being stimulus, physiology, and behavior. The first 2 steps, distal stimulus and proximal stimulus, referring to the actual light that is reflected from the object in vision and its representation in the sense organ, that is in the sensory receptor. These two are related to stimulus. We are grouping them into one category called stimulus. Now, step 3 is the reasons behind the representation. Remember, the receptors are the structures because of some chemical changes within them. That's the reason why the information is captured. So, step 3 is like a reason behind step 2, meaning that proximal stimulus is formed because of the chemical changes that occur in the receptor. That's why that's step 3. These changes that occur are the first transformations, right? But these representation that is formed in your receptors, these electrical signal representations, they go through different layers of the retina in your eyes, and also within the brain it undergoes changes. All these changes that are happening in neurons. Why are these neurons? Remember, receptor is also a neuron, and everything else, the information it encounters, also are neurons at different parts of our system, either in our eyes, if these are different layers of the retina, or the brain, which again is made of neurons. We call all these processes together as neural processing. So that's the physiology. Then, if you look at the perception, recognition, action, that's happening at the higher levels of your brain, and they are about your conscious experience of the external world. Perception, you know that you're experiencing something in the given medium. Recognition, you not only know that you're experiencing something, but you also know what you're experiencing. That is recognition. Once you've recognized it, you take a proper action. So, all these are higher level behavior related. So, what is the benefit of putting these 7 steps into 3 categories? Well, one benefit is now you can try to understand the relationship between these 3 categories to measure perceptual processes. That's exactly what we're going to do in this chapter. As you can see, there are 3 different connections that are formed here because of these categorization. What are they? A represents the relationship between the stimulus in the external world and your conscious experience of it. Could be perception, recognition, or action. This is one way of measuring perceptual processes. And how do you do this? Well, you manipulate stimulus and measure the resulting behavior in the individual. This is also called psychophysics. Why is it important? Well, this was the first way researchers started measuring perceptual processes. The interesting thing is, this measure was not only the first measure, it was the measure that researchers adapted even before there was psychology. In fact, it was in 1850s that 2 researchers by name Fechner and Weber, they developed ways of studying stimuli like light to understand the relationship between the properties of the stimulus and our experience of it. They were actually physicists. So you can see how these ways of measuring the relationship between stimuli and behavior, despite the fact that they were related to physicists, eventually laid foundations to psychology, which wasn't formed till late 1800s. The second way of measuring perceptual processes is B, which is specific to find the relationship between stimulus and the physiological changes that it results in. In A, we're manipulating the stimulus like light and measured the behavior by asking the participant. Whereas in B, you don't ask the participant, you measure the resulting electrical activity. The question is, why do you do that? Why do you need to do that? Well, you need to understand the logic here. What we're basically saying is stimulus results in changes in our neurons. These changes in the neurons are the reason why you experience the external world. So what you're trying to do now is to find the connections. Is stimulus actually causing the changes in the neurons? Number one. Number two, is changes in the neurons the reason why you experience the external world? The second part is difficult, remember? You cannot manipulate the neurons to see if that is the reason why you're experiencing the external world. For now, though, I want you to think about how stimulus can result in physiological changes in our neurons and how stimulus can result in our behavior. If we know these two, we can infer that the reason why you're experiencing the external world is because of the physiological changes. And that's exactly how we measure it. Now, it will all make sense once we go through an example. And the example we're going to use here is called oblique effect. Oblique effect refers to a phenomenon we experience vertically and horizontally oriented objects better than objects that are oriented in other angles, oblique objects. That's why it's called oblique effect. Again, horizontal and vertically oriented objects are experienced better than objects that are experienced in any other angles, hence oblique effect. Why so? We'll also talk about it later on. For now, let's see how this is measured. First, we'll look at the oblique effect through A, meaning we'll finding the relationship between stimulus and behavior through oblique effect. Then we do the same physiologically using oblique effect, and we'll talk about how we do this in the next slide. So in these experiments, you see the black and white stripes here. These black and white stripes are also called gratings. These gratings can be of different orientations. As you can see here, the grating here is in vertical orientation, the grating here is horizontal orientation, and the grating here is in oblique orientation. That's one part of it. And we're trying to see if participants experience vertical lines and horizontal lines better than lines in other orientations. That's our idea. Here, you have another construct, grating acuity. What is grating acuity? Grating acuity is your ability to identify the thinnest grades. Just imagine if you're presented with vertical lines with wider stripes and asked you to identify the orientation, you would say vertical. It becomes thinner. If we keep reducing the width of these gratings, there would be a point where you no longer be able to identify the orientation of the grading because it looks something like this. So we measure thus thinnest point beyond which a participant can determine the orientation or the stripe. That thinnest width would be the grading acuity. In oblique effect experiments, the materials used are called gratings. These are black and white strips of different widths. Participants would state the gratings orientation as gratings become thinner and thinner till they can't identify, and they measure the grating acuity, which is the thinnest identifiable width for a given orientation. They do it for vertical, horizontal, and for oblique to see which orientation has the finest grating acuity. What they manipulated was the orientation, and they measured grating acuity, and what they found was that the grating acuity was higher for horizontal and vertical gratings as opposed to oblique. Meaning, if the gratings were horizontal or vertical, participants were able to go to a point that were very thin and still say it is vertical or horizontal. But if it were oblique, they were not, meaning that your acuity to identify smallest lines was better when the lines are presented in vertical or horizontal, not in oblique angles. This gives us oblique effect. Let's move on. The animal we have here is a ferret, and the physiological measure that is used is called optical brain imaging. The participant, meaning ferret, is presented with gratings, horizontal, vertical, or oblique, and the brain activity in the ferret is measured through optical brain imaging, using light to measure the electrical activity of the brain. And the result was that the brain activity in the ferret was higher for horizontal and vertical gratings when compared to oblique. This shows how we use B to find the relationship between the stimulus and physiological changes. Now, what about C? How do you measure C? Well, as I said, it is difficult to manipulate the brain and measure the experience. We can do that under circumstances. We'll talk about that later. One way we do is by inferring C. How would you infer C by establishing A and B? Establishing A and B would be to establish it in the same experiment. In other words, you take the same materials and everything is same as experiment 1, where you measured A, grating acuity, by presenting participants with gratings of different orientations that would reduce in width. and measured the grading acuity for different angles. In addition to that, the brain activity is measured, meaning they're doing this task in a machine like fMRI, which measures the brain activity. What is manipulated here is orientation, just like experiment 1. And what they found in this experiment was that not only the acuity was better for horizontal and vertical lines when compared to other angles, the brain activity was also better for horizontal and vertical lines when compared to other angles. A and B is established. You can also infer that physiological changes is why most likely you get the experience. Again, there's a problem with this inference, which we'll talk about later when we talk about the big problem. I'll pause here. It's been a long video. In the next video, we'll talk about the specific ways of measuring A and the historic significance of it and the classical methods that Fechner had developed. Stay tuned. I'll see you in the next video.